Oh, hi! We didn't see you there. How are you today?
I'm Lauren.
I'm Tori.
I'm Kate.
And we're going to teach you about
DNA replication,
cell cycle,
and mitosis.
The first thing we're going to talk about is DNA replication. DNA replication is the process of making two identical strands of DNA from one original strand. DNA replication is a semi-conservative replication, meaning it uses part of the original strand. DNA is made of two strands, and in replication, the strands split apart and two new strands form with the old strands.
To split the DNA helices, unwind at the double helix, unzipping it. Single-strand binding proteins stabilize the unwound DNA. The next step is topoisomerase, which cuts the twisted DNA to relieve pressure. Then we come across base pairing, which is when the free nucleotides in the nucleus start bonding with the base pairs of the original DNA molecule. This happens by the work of DNA polymerase.
The leading strand is 5' to 3', so it is synthesized by DNA polymerase 3. The lagging strand runs 3' to 5', so primase synthesizes a short RNA primer, which gets extended by DNA polymerase to form an Okazaki fragment. After the RNA primer is replaced by DNA polymerase 1, DNA ligase joins the Okazaki fragment to the growing strand. We then have two new DNA strands with one strand of new and one strand of old DNA.
Next, we are going to talk about the cell cycle. The cell cycle is a accumulation of steps that DNA goes through throughout its lifespan. It is also the sequence of both growth and division. First, DNA untwists the superchromatin, which is DNA wrapped around proteins. The proteins are also known as histones. The next step of the cell cycle is the unzipping of DNA. In other words, when a DNA molecule separates at the base pairs by the work of enzymes or helicase.
Mitosis is when a cell in its nucleus divides in 4 phases: prophase, metaphase, anaphase, and telophase. You can remember the phases with the acronym Penguins May Attack Today. Prophase is a stage before the actual process of mitosis. DNA forms chromosomes, proteins that carries the genes and functions of hereditary functions in the cell. The membrane and nucleus are still visible, and the centrioles migrate to the opposite sides of the cell.
Metaphase means after. Spindle fibers attach to the centromeres of chromosomes, and they align at the equatorial plate in the middle.
Anaphase means in between. The spindle fibers shorten, causing the centromeres to split. The sister chromatids are pulled behind the centromeres.
Telophase means at the end. The chromosomes reach the opposite poles, the nuclear envelope reforms, and the spindle fiber disintegrates. The result is 2 identical daughter cells.
We hope you have a better understanding of
DNA replication,
Cell cycle.
and mitosis. Thanks for watching.